Hello, everybody. Good morning. Day 3 of the conference. Welcome aboard again. We'll begin with an outstanding, I would say, plenary session run by Becki Lawson. Becki, come on stage, please. I'm positively sure that it will be a very interesting talk, because very often we are, when we see that the population decreases of our species, the species we study, very often we think, well, habitat changes, climate change, etc., etc. But there are other options in terms of facing the problems. So, Becki, the stage is yours. Thank you.
Okay, thanks very much. Right, let's see if we can work all of this. Good morning, everybody. Thanks very much for the invitation to be here. My name's Becki Lawson, and I'm a wildlife vet from the Zoological Society of London. And what I plan to do this morning is to start off introducing disease as a potential driver of wild bird population declines with some international examples. And then I'll explain how we use citizen science to conduct wild bird disease surveillance in Great Britain. And then I'll focus on 2 emerging infectious diseases that we've investigated over the past 10 to 15 years, illustrating the ways that we use the various data sets to try and investigate their epidemiology and to quantify their population level impact. So that's the plan. As has just been touched on, we're all familiar with habitat loss, degradation, climate change, pollution, overexploitation, invasive species. These are all really important drivers of biodiversity change. What have I just done? Maybe it wasn't me. But I think that there's still a gap. I think there's still an under-recognition of the potential role that disease can play in this process. So this study reviewed the IUCN Red Data List and focused on species that have become extinct since 1500. And they identified infectious disease as one of the contributory threats to that process for 4% of those extinct species. And for wild birds, this very much focused on island endemic species, which were vulnerable to incursion of novel pathogens, to which they were immunologically naive. And as I'm sure you're aware, the most important and frequent examples are from the Hawaiian avifauna threatened by malaria and pox with mosquito vector introductions. Disease is also recognized as a very important additive threat for critically endangered species. And a good example here would be the issues that citizen beak and feather disease caused by a circovirus has posed to species recovery projects, for example, for the echo parakeet and for the orange-bellied parrot. So disease is an issue when there are very few remaining individuals. There's a great deal of focus on emerging infectious diseases, and these are defined as being caused by novel pathogens or known pathogens in new hosts, in new locations, or at increasing prevalence. And in terms of infectious disease threats to global vertebrate biodiversity, there are 2 fungal pathogens which are really the most headline news, and these are Batrachochytridium dendrobatidus as a cause of amphibian chytridiomycosis. This recent study published last month estimates the extinctions of 90 amphibian species as a result of this disease emergence event. And the second would be Pseudogymnastus destructans as a cause of white nose syndrome in North American bats, leading to population collapse and the deaths of millions of bats. So these events really illustrate how disease can have a rapid and unpredictable effect on previously common species over a wide geographical span and within a short time frame. Much of that which I'm going to talk about focuses on infectious disease, but I just want to briefly remind everybody about the potential threats that non-infectious disease can pose. And for wild birds, these are 2 familiar, very important examples: the organochlorine pesticides that led to collapse of predatory bird of prey populations, particularly in the 60s and 70s, and then more recently, the impact of diclofenac on gyps vulture species in India and Nepal, this anti-inflammatory drug for livestock that had an unexpected consequence of being highly nephrotoxic when the vultures scavenged the carcasses of fallen livestock. There are multiple diseases that can lead to mass mortality events of wild birds. And for waterfowl, we might think of things like avian cholera, caused by Pasteurella multocida, a particular issue for North American waterfowl, or duck plague, duck viral enteritis, caused by annatid herpes virus, maybe avian botulism. There are different examples. And in seabird wrecks are not uncommon events with numerous causes, including starvation, but storm events, oil pollution, more recently PIB pollution and the like. So these mass mortality events of larger-bodied birds do occur, and in these situations what often happens is that people try and estimate the overall mortality, and then they review that in the light of the most recent regional population estimates for those species, bearing in mind their breeding ecology, and try and make some inference on the likely significance of that event. For birds of smaller body size, there have been a number of studies over the years trying to look at detection rates of carcass in the field and also rates of carcass loss due to scavenger activity, with a goal to moving towards extrapolating from the number of carcasses retrieved in the field to an estimate of the overall scale of the event. But I believe that to robustly identify and attempt to quantify population-level impacts, it's a team effort that requires direct integration of surveillance data sets with population monitoring data sets that are available with comparable continuous methodologies both before and after these disease emergence events. So that's the take-home message. And there are a number of examples where this has been done over recent decades, starting with the work of, I can't see where he is, but Wesley's here in the audience and presenting on Friday. So house finch conjunctivitis caused by Mycoplasma gallisepticum, a bacterium, emerged as an issue in the mid-1990s in the eastern US and caused these very clear external signs of disease in the birds. And Cornell, who already ran citizen science schemes, took this opportunity and asked people to report sightings of sick birds to them and then used that information to track the spread of the outbreak in time and space. And then looking at the Christmas bird count data for incursion of that disease in different regions, they were able to look at the historical trends and post that line of incursion, document a decline in the order of up to 60% population decline within just a few years. Yeah. Okay. The second important example would be the introduction of West Nile virus into New York State in 1999. So West Nile is a flavivirus spread through mosquito bites with a wild bird reservoir cycle, but the potential for spillover to cause serious disease in people, horses, and other hosts. American crows are highly susceptible to this virus. In fact, they were used as a sentinel species in the early years. So in this plot, the group used the BBS counts, and they looked at the historical trajectory of the American crow populations, which was increasing, and then across the states, in different areas, they looked subsequent to the orange line of incursion and quantified a decline of that species up to around a level of about 45%. Next, EBCC virus. I'm going to whisk through this because we're going to hear about it in a lot more detail in the next presentation. But just to say, also in Europe, this important flavivirus originating from Africa that's particularly well known as a cause of blackbird mortality. And in this study, the group tried to appraise the impact of an outbreak that occurred around 2011 in Germany. And using climate data, they examined areas of EBCC virus suitable in green, and unsuitable habitat in white, thinking about the mosquito vectors. And then they modelled those population trends and showed that subsequent to the disease emergence in 2011, there was a 15% reduction in the blackbird population in the Yasutu virus suitable over unsuitable habitats. So they used that to try to quantify the impact of the disease on that species. So those are some examples. Now I'm going to move to the work that we conduct in Britain using citizen science. I believe that in this context, the approach is beneficial to both citizens and the scientists. Not only from the public's point of view is it an opportunity to engage with nature, contribute to science, but it's also a means to access information on the common conditions that affect our wild animals and get advice on habitat management to help try and prevent or control disease outbreaks, and also how to safeguard public health and companion animal health. From our point of view, we appeal to members of the public to let us know when they see signs of ill health in their wildlife locally. And that means that we undertake surveillance across the year, throughout the country, in both private and publicly owned land. And that would financially be otherwise completely impossible. So hopefully it's a win-win. In recent decades, there's been a real growing popularity in interest for wildlife-friendly gardening practices, and a common element of that is the provision of supplementary food for garden birds. And this is now done sometimes on a large scale and typically year-round rather than during the winter months only. And these two factors mean that there's a close interface between garden birds and the public, and that gives us the opportunity to undertake surveillance. And this began in the early 1990s when members of the public saw dead birds around their feeding stations. They were motivated to find out what was going on, and so they contacted the vets at London Zoo, who helped by conducting post-mortem examinations. And that enables us to reach a diagnosis, but these ad hoc or opportunistic submissions are very valuable, but they are limited, and we need to be aware of these restrictions. So we have no control over observer effort in time and space. And that means that if there's a regional media piece, there might logically be an increase in reports of that condition in that area. And if we weren't aware of that, we might misinterpret that as a localized outbreak. People are much more likely to get in contact and tell us about issues that affect the species which they view as charismatic, as opposed to those which are less positively viewed. So we really need to be sensible about what we're trying to achieve. There's bias. People tell us about events that involve multiple birds or where they perceive the cause to be unknown. There's a time delay, always a lag, which means that it can be quite difficult to obtain specimens that are well preserved for examination. But most importantly, the disease surveillance information in isolation can't enable us to infer whether there's a population-level impact. So we can learn a lot about the diseases that affect wild birds, but our conclusions are limited in scope. And that's what led us in 2005 to set up the Garden Bird Health Initiative, bringing together contributions from lots of stakeholder organisations, so really collaborative from the outset, in an effort to set up a national wild bird disease surveillance scheme. And we continued with two independent but complementary surveillance methods. The ad hoc reports mean that any member of the public can get in touch, so we have maximum observer effort. But we combined that with the first example of systematic surveillance by collaborating with the BTO's Garden Birdwatch scheme. So this has a membership and around 13,000 participants across the country who provide information every week throughout the year on the wildlife that they observe in their gardens. And what we did was recruit a subset of these people and ask them to let us know whether or not they saw signs of ill health in their local wildlife. And that gave us a structured negative sightings data set that we could use to control for reporting bias and do more robust analysis. Over the 25-year period, as you'd imagine, there's been a real evolution in reporting modalities, and I'm pleased to say we went online around 5 years ago. We conduct scanning surveillance or general surveillance, and that means that we are interested in both infectious and non-infectious disease as a threat, as a cause of death, but also in underlying conditions. So it's broad in scope, and over that time we've developed a number of set protocols and definitions that enable us to compare like with like in time and space. But gardens are valuable habitats for many species, and so in 2013 we launched the Garden Wildlife Health Project, which brought together both the garden bird and the amphibian disease surveillance which we've been conducting, and added in terrestrial reptiles and hedgehogs for the first time. And we continue to work in partnership with the BTO and the RSPB and FrogLife, and we're funded by government and charitable foundations. So that systematic surveillance continues now through the online reporting portal for Garden Birdwatch, where they have a single health tab whereby people are invited, of everything I saw this week, did I see any signs of ill health? Check one box, and that gives us our negative sightings data set, so very quick and easy. If anybody sees the problem, they are forwarded to our website, all free to use, free to report, examinations supported by the project. They register their site, which can but doesn't have to be a garden, and then they can report any number of disease incidents, giving us information on the date, the species involved, the numbers of individuals involved, and in the event that they found a freshly dead carcass and they'd be willing to liaise with the project vet to arrange its submission, they give a contact detail, that fires an email to the vet, they call them and explore that possibility. And people also are invited to describe what they've seen with free text fields and can upload photos. And these can be really helpful. So basic examples, things like differentiating exoded ticks from avian pox lesions. Quite straightforward. The most common thing we have reported to us would be chaffinch leg lesions caused by pneumococcal mites, mange, or chaffinch papillomavirus, or a combination of the two. We're on the lookout for things like overgrown beaks, something we have infrequently reported, but we're aware of the emergence of avian keratin disorder as a frequent cause of overgrown beaks in North America, so we're horizon scanning for early detection of that as a potential threat. But oftentimes, the birds are just fluffed up and lethargic. They're telling us that they're systemically ill, but without further investigation, we can't reach any robust diagnosis. And so through both of these streams, we solicit specimens for examination. We're mindful of the potential for zoonotic infections, those that can affect people, so we give appropriate public health guidance. And the examinations are conducted at the labs at ZSL London Zoo, using the suite of diagnostic tests that you would do with any species. And we have archives of tens of thousands now of formalin and fixed parasite culture collections and the like, which are a really invaluable repository for future research focused on disease or elements of more ecological research. We're a citizen science project, so we take time to report back findings to each submitter, particularly highlighting if there are ways that they might manage their habitats to try and prevent problems in the future, or if there are public or companion animal health issues that might relate to our diagnosis. The Garden Birdwatch information is used to generate relative abundance estimates, and that means we have achieved direct integration of the disease surveillance with the population monitoring. And our goal is to try to bring this together, looking at the information we have and giving evidence-based guidance. So we're an academic institute, so the first port of call are peer-reviewed publications, and we opt for open access journals wherever we can do, because the public have helped us collect the information and we want to ensure that they can access all of the details. We have an interactive mapping tool on the website, which means that people can get a feel of what's happening in their region as well as in their garden. We have a library of around 30 fact sheets on the common or important conditions that affect the species groups that we work with, and these are a digestible, accessible synopsis for members of the public and wildlife rehabilitators to use. We are part of the government's network for wildlife disease surveillance, so we contribute to open access reports with recent findings each quarter that are published on the government's website. We do electronic newsletters to reporters a couple of times a year. We use social media extensively, but to raise awareness of findings or seasonal issues, not to try and capture reports, because that can lead to very poor quality information. We distill those peer-reviewed findings into popular science, disseminated through collaboration with many different animal welfare and conservation NGOs, and we spend a lot of time at public outreach events trying to raise the profile and awareness of disease as a potential issue for wild animal welfare and conservation. So that's the what and how we do bit. Now going to move on to the first example, which is the emergence of finch trichomoniasis. And with this slide, try and emphasize how collaborative the work has been already. Many different organizations involved. So the background here is that Trichomonas gallinae is a protozoal parasite that's been known for hundreds of years to cause disease in pigeons, doves, and birds of prey. It causes these lesions to develop at the back of the throat and the oropharynx, which interfere with the bird's ability to swallow, so they regurgitate food, they drool saliva, and the parasite is spread in fresh saliva. It's killed by desiccation, it can't survive for long in the environment. So pigeons and doves would become infected if the adults feed one another during courtship, if the adults feed their young with crop milk, or potentially at shared food and water sources. And that's where supplementary feeding stations may come to mind. And birds of prey are exposed when they consume infected hosts. So that's the typical known situation. But then in 2005, in Britain, we first started to see atypical seasonal mortality, primarily of greenfinches, with chaffinch being the second most commonly affected species. So the first step were the traditional veterinary tests required to reach a diagnosis. And again, at post-mortem examination, these birds had very clear upper alimentary tract lesions from which we were able to isolate the parasite and use both morphological and molecular techniques to confirm its identity as T. gallinae. So this was an emerging disease in a novel host. But the following year, in 2006, we saw epidemic mortality in these same species. Now, the RSPB Wildlife Inquiries Unit team had collected reports from members of the public where the history suggested that a disease may be involved with a comparable level of intensity of surveillance since 2001. And in the years 2001 to 4, the winter is the modal season, and that's accounted for by passerine salmonellosis, so entirely different disease. But that you'll see in 2005, when finch trich was first seen, autumn becomes the modal season, and then in 2006, there's a 5-fold spike in the number of reports. And it's really important to know that that wasn't as a result of a media appeal, but that that was driven by the scale of the outbreak itself. The greenfinch remains the most commonly affected or commonly diagnosed at post-mortem examination species. Chaffinch second most commonly, but a really wide range of passerines are susceptible to the disease. Over the first couple of years, we looked at the number of dead birds that people had found who had reported to us. 40% of people had found in excess of 6 dead finches. So we had not only a spike in the number of reports, but an indication of quite large-scale mortality at each site. That led to national media attention for animal welfare reasons, but we wanted to look at whether or not there may be evidence for a population-level decline. So the first step was to use the Garden Birdwatch dataset. So here we have in the grey lines the plots for the decade preceding this disease emergence event. So you can see a typical seasonal peak in spring and a dip in autumn. And in 2006, the red line is within the historical trend until it diverges and remains depressed. So this was evidence for a UK garden greenfinch data set of a significant decline that was contemporaneous with the onset of epidemic mortality. But we wanted to look in more detail at these population trends before and after the emergence, comparing trends in high and low disease regions, and using the dunnock as a control species to try to check whether any other environmental factor might explain the observed trends. So we went through all of the reports and created a rule based on species, time of year, signs, and including post-mortem data where available to identify trike incidents. And then we looked at those two data sets in turn. So we started with the opportunistic information, and here we have a total number of incidents by county stratified by the number of human households, so we could try and control for observer effort. And you can see in that first year that there's a very clear peak in the western counties of England. And we then created these high, intermediate and low county groupings. But before we used those for analyses, we went to our independent systematic data set to double check whether the trends were the same. So these tiny yellow dots are monitored sites. The red dots are sites in which finch disease was observed. And you can see that 1 in 10 in the high and none in the low region reported these problems. So we had an independent assurance that these regional county groupings were robust for the analysis. Then we moved towards the BBS data. And we have high, intermediate and low regions. With the greenfinch, the species we predict would be hardest hit. The chaffinch, the second most commonly affected species, and the dunnock are control. These box whisker plots represent the extent of interannual variation for the decade preceding the disease emergence event. And then the black circle is what happens after the first year of outbreak. And so we see a decline of around a third of the greenfinch and around a fifth of the chaffinch population in the high region, no significant declines in intermediate or low, and no changes in our control species. So this is consistent with a disease-mediated population decline hypothesis. And if we then bring things forward almost to date, looking now at the BBS data for the UK as a whole, you can see a pretty alarming, significant annual ongoing population decline for the greenfinch. In fact, a recent study by Stanbury et al. Used the IUCN extinction risk criteria on a national basis for British bird populations and concluded that the extent and scale of the decline would be sufficient to list the British greenfinch as endangered under those criteria. So it really demonstrates how dramatic these events can be. So as I've said, in 2006, the focus was on the west of England, and in 2007, the epidemic spread eastward, we think through greenfinch movement within the country. And then in 2008, further east into Fennoscandia, where disease incidents were first seen in the same species groups. So the red dots here represent the index sites, and the paler county groupings are where reports were received. Important to see that they're not clustered around the coastline of Norway, and we'll come back to that. So, how might this have occurred? As I've said, it's a very delicate parasite, so it's not amenable to long-distance transport, and there were no reports of trike in other species in that area. So we wanted to look at bird migration as the potential route of spread. And to do that, we worked and looked at birds ringed within Great Britain and subsequently recovered overseas. And for the greenfinch in black, those international recoveries were predominantly around the coastline of Norway and France. But for the chaffinch, there's wholesale overlap in the areas where the disease emerged in Fennoscandia. And if we look at other species in which trike is frequently diagnosed, we see no similar spatial overlap. And so our hypothesis is that it's most likely migratory chaffinches that left their wintering grounds to breed in the summer of 2008, taking the parasite with them. And that hypothesis is strengthened by the fact that when they return, they take a path along the coastline of mainland Europe, and then outbreaks were subsequently seen in Germany, the Netherlands, and Denmark. It's reached as far as Austria and Slovenia, 2012, 2013, And these are really widespread species, so there's an important opportunity for a wider scale collaboration here. The declines of greenfinches is something that's been highlighted in an EBCC leaflet, and I really hope there's going to be opportunity to collaboratively investigate the extent of the declines and see if we can elucidate any factors that have influenced how these have varied over the species range. So as I said, pigeons and doves are considered the reservoir, with typical spread to birds of prey. Our hypothesis is that it's a single clonal strain of the parasite that's spilled over from pigeons or doves in Britain into finches, but that the epidemic has been sustained predominantly finch to finch since then, with potential secondary spillover to bird of prey populations. There's lots of work done on the molecular aspects of the parasite to try and elucidate these pathways. In terms of birds of prey at risk, if you were going to look at one of higher risk, it might be the sparrowhawk, chiefly reliant on small passerine prey and a common visitor to garden habitats. So it's interesting to note that we have seen, since the orange line, since the onset of finch strike, we have seen a downturn in sparrowhawk numbers. At this point, we don't have data to really ascribe causality here. It's a hypothesis, but it remains a potential explanation. So just to say, why did this event occur? We don't know, but there are a number of options. The pigeon populations had been increasing because of arable crop management. Greenfinch populations had been increasing as a result of supplementary feeding. And as I said, there's this shift to year-round feeding, which means that there's an opportunity for larger numbers of birds to congregate throughout the year, which might predispose to spillover events occurring. But it could also be that there's been a virulent strain of the parasite, perhaps with a larger and extended host range that has emerged. And so we continue to work on these aspects to try and answer that question. And then, second and much more briefly, moving on to parody pox, a totally different scenario, but again, a very collaborative piece of work. So avian pox, many different strains, loads of bird species susceptible. It's a DNA virus. It's pretty stable in the environment this time, and it's spread through contact between birds, particularly if the skin is abraded, or through biting insect vectors such as mosquitoes. It causes lesions most commonly in the skin, around the eyes, around the beaks, on the feet. The lesions can be mild, the birds can recover, but if they interfere with vision, feeding, locomotion, then they can have a serious impact on individual bird health. In Britain, pox is considered endemic in at least these species. Reports since the 50s, typically individual birds affected, and the lesions are often mild. So here are some examples. This is an incidental skin lesion in a feral pigeon. Oh, what have I done? And moderate severity lesions in a dunnock. So again, that's the normal background situation, but in 2006, in southeast England, we had the index case of a really severe form of skin disease in great tits. So over a 4-year period, we received over 200 reports of these birds. 90% of them included great tits. 75% of these incidents had multiple birds affected. 20% involved blue tits. So pretty striking lesions. First step again, traditional veterinary tests to reach a diagnosis, which with this condition are pretty straightforward. Under the microscope, we can see these pink bodies, which are viral inclusion bodies called Bollinger bodies, which are considered pathognomonic or diagnostic for this disease, and from them we were able to isolate the virus and confirm that. But these are one of our most ubiquitous, best studied, numerous species, really striking condition. We were confident it was new, but the question was, where had it come from? And to answer that question, we went to our sample archives and picked out skin lesions from the birds in which that infection was considered endemic, and found evidence for multiple circulating strains of the virus that were associated with different species. And when we looked at the great tit, we found that that was a separate virus sequence to all other birds apart from the dunnock. So could this represent a spillover event from a dunnock to a great tit in Great Britain, or was there an alternative explanation? And to look at that, it's important to be aware that parody pox has been sporadically reported in Scandinavia since at least the 50s. And then Ivan Literak et al. Published a cluster of reports in Austria, Slovenia, Czech Republic, Germany in the mid-2000s, and then we had our index case. So given this link in time and space, this perhaps looked much more like an incursion event of a novel strain of the virus. And so to look at that in more detail, we examined the distribution of the parody pox reports we'd received and saw that they were really closely clustered to mainland, proximity to mainland Europe, and there was clear evidence of an epidemic spread northward and westward. So very reminiscent of a point source introduction followed by an outbreak. So how might that have occurred? Again, could it be due to bird movement? Well, as I'm sure you all know, but I didn't, great tits are very sedentary in Britain with few international exchanges, so bird movement is a much less likely explanation here, and instead mosquito movement might be the reason. And in a similar way, we continue to work with virologists to try and learn more about the genetics of the virus and why the disease may be so severe. Just to say that there was a parody pox outbreak in a very intensively studied population of tits in Whiteham Woods that the University of Oxford has monitored since the 1960s. And this gave us the opportunity to look in detail at an outbreak. Lots of numbers here. We can ignore all of them apart from two. So over a 2-year period, around 2,000 birds were captured. For great tits, the average prevalence was 5%. So 1 in 20 birds had signs of skin lesions. The blue tit was 100-fold less commonly affected, and these birds moved together in mixed groups. So that tells us that the great tit's probably more susceptible rather than more frequently exposed. Then these birds are ringed, so Oxford could do multi-state mark recapture work to demonstrate a very significant adverse impact on individual bird survival. That was most marked for juvenile birds. And they're also monitoring the nest boxes so they could look at impacts on reproductive output with no changes at the early stage, but a really severe decline in the number of young reared to independence. So with that detailed information, we wondered whether we were in a position to make some predictions about whether we might see a population-level impact. So again, Oxford led creation of an age-structured model, which did predict that disease-mediated decline could occur, but that probably wouldn't kick in until prevalence exceeded around 8%. We'd seen 5% in the field, so we felt it was perhaps unlikely to occur. But we continued to monitor, and the UK BBS trends, interestingly, subsequent to the arrival of great tit pox, do show a change with a plateau followed by a drop. And so to look at that in more detail, we went back to our spread of disease outbreaks, moving in the early years from red to purple to blue, so into Northern England and Scotland more recently. Then we used the BBS data again. So the plots on the right, and I think this is going to be difficult to see, are great tits, and on the left are blue tits. The black lines are what happens for the decade before the disease emergence, and the grey lines are what happened the decade afterwards. And the take-home messages are that in England, for great tit, there is a significant decline, but that does not match the magnitude that we would predict based on our knowledge of the south to north disease progression. And the blue tit has non-significant declines in that same area, which we know to be much less frequently affected. So there are signals here that are consistent with disease-mediated decline, but we think it's likely that climate, food, other factors are also influencing these populations. So it's a more complicated and subtle picture. So that is finishing off that story. I just finally want to show this slide to illustrate the use of other data sets to look at disease impact on wild birds. This is across states looking at bird banding information for about 50 species, focusing on adult survival and looking at the impacts of West Nile, whether it was a temporary population dip or a more sustained decline. So it illustrates how the different data sets that are available can be used to appraise disease impact. And my final slide is to say that I hope that this illustrates how vets and disease ecologists working together with ornithologists using all of these data sets can enable us to start to look at the role that disease can play as an influencer of wild bird population trends. And I really think that there's an opportunity to work more closely with ringers, focusing or extending the range of surveillance away from these peridomestic species alone into other species, and also working with conditions typically associated with morbidity but not necessarily mortality. So I hope that there's a real opportunity for collaboration in the future. I want to thank co-authors, particularly Rob Robinson, who's here today, partner organisations, funders, but most importantly, the members of the public who take their time to report to us because they care about wild animal welfare and conservation. Thanks very much.
Just one quick question. I'm sure you have lots of questions, but Becki will stay with us today, so we will have the opportunity to raise more questions. Yes, please.
Thank you very much. I'm Åke Lindström, Lund University, Sweden. Would you have any examples of the opposite happening, where a species would sort of escape from a disease and actually increase in numbers?
No. There are examples of kind of positive response in populations following threat removal. And the examples there I think of would be more non-infectious diseases. So there's quite a neat study recently on the response of mute swan populations following the ban of anglers' weights and lead as an issue. And they've looked at various data sets to look at evidence for that being a species recovery. So perhaps that's easier to achieve with non-infectious disease. I'm sure there are many examples. By the time we've got to coffee break, I'll have thought of them. Thanks for the question.
Thanks,
Is that okay? Thanks, everybody.
On population dynamics. Okay, sorry.
Please.
Sorry. Good morning, everyone. So I'm Danai, and I'll be moderating the next session entitled Population Dynamics. We're going up to the coffee break at 10:15. Whoever needs to move, better move now. We are starting with Mark Hermans from Natuurpunt, from Belgium, on recent impact on ESUTU virus on birds in Belgium. And you have all, just to remind you that speakers have 12 minutes and 3 minutes for questions. Thank you very much.
Good morning. I will be talking about what appears to be the impact of ESUTU virus on a number of species in Belgium. ESUTU virus is an encephalitic flavivirus transferred by mosquitoes. So it's closely related to West Nile virus or to yellow fever. That's pretty nasty stuff, actually.